So I have been given the topic today to talk about research translation in vaccine development, and what I have chosen to use today is the story that I've been involved with Professor Yati and Pakitov around the rotavirus vaccine and the development of the rotavirus vaccine. The story is not dissimilar to the story of other vaccines, but they all have their own little nuances and quirks. So translation is the cycle then between the bench, what happens in the laboratory, what happens at the bedside in the clinic, and what happens in the community, the public health aspect of what we do. And it's those three aspects, the continuing cycle of information feeding back from the laboratory to the clinic to the community, is what drives a lot of vaccine development and drug development. And I think it's a very appropriate driver for development of new products. There is problems though, because in academia, in the laboratory, and in situations where we find ourselves, the academia, we have a lot of information, we learn a lot about specific drugs, specific diseases in the laboratory, but often there's a big gap between using the information that we develop in universities and in research institutes to develop a product that's going to be able to be used in the clinical setting and in the public health setting. That's called crossing the valley of death. There are many products, many good ideas that never make it out of the laboratory and into a product, and that's a great shame. How to resolve this issue, stop babies dying and being very sick from diarrhoea and dehydration. We know now that diarrhoea is a leading cause of death in children less than 5 years. About 800,000 children worldwide die each year because of diarrhoea. There are a number of other important causes, neonatal causes, of which diarrhoea will be part of the neonatal deaths in the first month of life, but it's sometimes very hard to identify the cause of death in very, very young babies. So it's a very important big sector to really, if you're looking at improving MDG goals, improving welfare of children in the world and in developing and developed countries, to attack the issues of pneumonia and diarrhoea, the big killers. Sometimes it's easy to get overwhelmed with the big four: AIDS, TB, meningitis, and so forth. But for children under 5, diarrhoea and pneumonia are the big killers we want to address in particular. And not only do children die of diarrhoea, but also it has have important feed-on effects. Approximately 1 in 5 children under the age of 2 who suffer an episode of moderate to severe diarrhoea each year. These children are 8.5 times more likely to die within 2 months of having a diarrheal episode, usually after they leave hospital. This is particularly in developing countries. So something about the diarrhoea, malnutrition as a result of that, and reducing their resilience to other conditions sets off a cycle for many of these children. And children who survive a severe episode of diarrhoea are more likely to be stunted compared to their peers over the same 2-month period. And that also reflects the time they need to recover from that diarrheal episode, to repair the gut, to improve their nutritional status, and to, at this important time of growth, to be able to sustain growth. So diarrhoea is an important contribution to stunting and poor growth in countries where diarrhoea prevalence is very high. And we're starting to realise it's not just deaths from diarrhoea, it's not just hospitalisations from diarrhoea, but there are these other important morbidity aspects. We haven't even gone into consideration here the neurodevelopmental aspects of having malnutrition and severe disease in very critical times of development in the first 2 years of life. But that's likely to be a significant factor in terms of the consequence of diarrhoea disease as well. It was this trying to understand what causes severe diarrhoea in children in developing countries that promoted or prompted Ruth Bishop, who is a good friend to the team here at UGM, to write down this clinical protocol. And the protocol actually says because many of the cases of diarrhoea back in the 1950s and 60s and even the early 70s, the majority of cases of diarrhoea we couldn't pick up a bacterial cause. And a lot of them were called idiopathic diarrhoea. So going into the laboratory, Ruth Bishop, who was a microbiologist, took the task to try and find out what the cause of the majority of cases of diarrhoea are presenting to the Children's Hospital in Melbourne, where, as I say, 80% of cases of diarrhoea, we did not know the cause, despite extensive bacterial culture. So just like you when you're starting off your research projects, you write your aim. This is her original aim, and this is the background with all the crossing outs. We didn't have computers in that time. And that was her original study. And this is the study that we recruited at that time, or she and Professor Graham Barnes and the team at the gastroenterologist at the Children's Hospital recruited infants who presented to the hospital with severe diarrhoea of unknown cause and did something called a duodenal capsule biopsy, where they placed a little capsule down into the stomach, because we didn't have endoscopy at that time, and took a little sample of the duodenal mucosa of those babies. And with that sample from the duodenal mucosa, Ruth would put it in her little ice chest and take it over to the University of Melbourne, where they had an electron microscope. And this is the first picture from the first baby in that study. And it was from, it doesn't project very well, but you can get the sense that there are these little dots. It's not presented particularly well there. And this was seen on the first view under the electron microscope of that first baby they did in that study. And this was something that hadn't been recognised before, and this is rotavirus. They identified these little features not seen before, called it originally or initially duovirus, and later it was called rotavirus because it has a rota type shape. When they worked then and collaborated with Professor Yati, looking at the stool of babies with diarrhoea, saw the same thing here in Indonesia. And Indonesia was one of the first countries to recognise that rotavirus was the cause of diarrhoea in Indonesia as well as in Australia. And subsequently, within the same year or two, all around the world, similar observations were made in Africa, in Asia, in Europe and America, that this rotavirus was common to all the children that, or many of the children that presented with severe diarrhoea. And this particular virus was then seen in animal studies, because it's much easier to infect animals than it is children and look at part of their bowel. But from the normal mucosa, which is, you would remember, is nice tall villi which help with the absorption of food and fluid, becomes quite stunted and damaged after an episode of rotavirus gastroenteritis. And so that is what causes the severe dehydration and watery diarrhoea, because you can't absorb from that surface and there are fluids leaking from it. Children don't die of the intestinal disease, they die of the dehydration caused by the injury. Intestinal mucosal cells reproduce very quickly, and babies can recover very well as long as they're supported through that period of dehydration. So from here we had a clinical observation. We knew that there was a big problem in the community, in the global community, with diarrhoea disease. There was, that went to the laboratory, an unusual and unique organism was identified, and then what? Then the team decided it was time to try, now we had an organism, to try and look at how we could prevent that organism. And this is back in the mid-1970s. Since that time, epidemiological studies through a variety of communities throughout the world have found that almost every child less than 5 is infected with rotavirus. It's called the democratic disease. It doesn't matter whether you're living in a village in Africa or in New York City, Yogyakarta or in Melbourne. Children less than 5 almost all have an episode of rotavirus infection. The difference is that children who have access to medical care are treated for their rehydration well and don't die. But children who have difficulty accessing medical care often do have much more severe disease that is not treated well with rehydration. It's the most common cause of diarrhoea worldwide in infants less than 11 months of age. And we know that from recent studies from Karen Kotlov and their team published in Lancet. And we also know that improvements in sanitation and drinking water do not adequately prevent rotavirus, not like the bacterial gastroenteritis. So a lot of it is spread by touching. It is very excreted in large amounts in the stool of babies that are infected, and it is aerosolised as well. In 2009, WHO recommended that all children around the world should receive a rotavirus vaccine for prevention. And this was really because there had been quite a lot of data collected on epidemiological studies around the world saying rotavirus was important and that we needed to find a cure and that vaccines were going to be the most likely, most effective way of preventing this problem. We talked about deaths a little bit. Hospitalisations are a huge problem, similarly, the outcome of a lot of those epidemiological studies. In terms of deaths, top 10 countries, Indonesia features in the top 10 countries where rotavirus causes death in children less than 5 years, with almost 10,000 deaths per year from the rotavirus, and certainly a lot of hospitalisations. So, we know a lot more now from 1975 about the rotavirus epidemiology and the importance of the rotavirus worldwide. We also know a lot more now back into the lab again around what rotavirus is, what it looks like, some of the serotypes that exist around the world. That's the little the variations in the type of rotavirus. These different strains vary around the world. This is very important for vaccine development because if you're developing a vaccine, you want the vaccine to target all of these serotypes if it's going to be a globally useful vaccine. It's not going to be much point to have a vaccine that's only works well in Malawi with this particular serotype. It won't work in other areas with different serotypes. And the other complexity is that rotavirus strains change over time. So it's not the same rotavirus strain each year. They vary with time, so that if you are creating a vaccine, you need to consider a vaccine that's going to be flexible and able to prevent against the range of serotypes presented in the community and changes over time, not only geographical changes. So this, from vaccine development, created a big challenge. But again, as I say, important information coming from the community, a lot of epidemiological studies that was going to drive the type of vaccine that could be developed and to be effective against this particular bug. The first rotavirus vaccine was developed by a man by the name of FK-KMK back at NIH in the early 1980s. And FK-KMK has also been famous for a number of other vaccines that have been developed, as well as discovery of norovirus as well. When you're starting to design a vaccine, it's important to think about when your target population might be most vulnerable to the disease you want to prevent, and what you know from nature about the type of protection that would be needed, that you need to develop when you develop a vaccine. What sort of protection, and from an immunological perspective, do you need to engender with the vaccine? And there's some hints from the natural history studies on normal wild-type rotavirus infection. One comes from Ruth Bishop and Graham Barnes back years ago, which looked at neonatal rotavirus infection. Neonatal rotaviruses, as I'll explain in a little bit, are quite unique rotaviruses that occur in newborn babies. And these neonatal rotaviruses create immunity against infection later on in life for the first 3 years, but don't cause disease in newborn babies. It infects very well the babies, but doesn't cause disease. Quite different to other rotaviruses that are disease-causing rotaviruses. So the information from that particular study is that if you were able to induce early infection, then you may induce immunity at the vulnerable period, which are little children, the less than 5 years, particularly less than 2 years. The most impressive immunity is developed after the first infection with rotavirus disease. So that if you have a baby who has a severe episode of rotavirus disease, it will be usually their first episode is the severe one, and naturally that produces infection for subsequent episodes. So it's less likely that you have a severe episode at your second, third, fourth or fifth. We are all exposed to rotavirus probably in the hospital here multiple times a year, but it's because we have engendered our own immunity means that we don't know we're infected. So you can't get rid of rotavirus from the environment, but what you need to do from the protection point of view is protect the babies from their first infection, so that when they get infected, they have the immunity in their body so that that infection does not cause severe disease. So the approach of the first rotavirus vaccine produced by FK-KMK took in the laboratory little samples of or parts of the rotavirus from human and also from rhesus from monkey, and combined them together to try and target those rotavirus serotypes that were most prevalent around the world. And this was developed as the RotaShield vaccine. The RotaShield vaccine went into the clinical trial development phase where, and you'll see the slide coming up a couple of times, where you have a promising vaccine candidate, but you need to make sure it's safe and effective in humans before you can actually license it and make it available to children or adults for that matter. And there is a series of phase 1 studies, which we'll talk about in a minute, phase 2, phase 3. Phase 1 studies are really to focus to make sure that the vaccine is safe. So it's well tolerated. It doesn't look at anything usually around immunity, but it's just well tolerated, and that there aren't any major issues of concern before you, and it's usually very small numbers in phase 1 studies, and quite specific, single dose, quite specific outcomes. Phase 2 studies are usually looking at safety and the immune response produced by the vaccine, so that you're not particularly focusing on protection of the vaccine, but you want to know that some marker that the vaccine is promoting some immune response, which you think will prove predictive of whether there is going to be protection. Phase 3 studies are usually the largest studies, and they're focused on to make sure that vaccine not only is safer in a larger population and well tolerated, but it's whether it offers the protection you think it should provide, and that gives you, it's called an efficacy study usually. This is what the regulators will need to give a licence for a vaccine to be produced, and before it can be introduced into a country or a region or globally. The next important part is that once the vaccine is introduced into the community, there is important what we call post-marketing surveillance or post-licensure surveillance, where you see how well it performs in the real world, outside the clinical trial setting, usually in a big number of patients or subjects, in the way you would have it in a normal introduction and implementation, and so that you would look at how effective the vaccine is, and if there are any very rare side effects that you have not been able to pick up in the clinical trial setting. All vaccines and most drugs will go through this process of development. So the regulator here, from the Indonesian context, and usually each country has their own regulator to introduce a vaccine. In order for a vaccine to be available for WHO, it has to go through another level of regulatory review by WHO, and that's called WHO pre-qualification, and that looks at how appropriate that vaccine is to be delivered into a developing country setting and purchased for Gavi-eligible countries or UNICEF or the variety of global vaccine procurement agencies. And in particular, that looks at issues around the cold chain, issue around bulk, issue about volume, those things that are important in successful implementation, particularly in resource-poor countries which have a difficulty with refrigeration or transportation and so forth. So getting back to the rotavirus vaccine story, the first rotavirus vaccine, RotaShield, was, as I mentioned, a human rhesus, had 4 different parts to it, called a tetravalent reassortant, produced in the laboratory, was licensed in the US in 1998, introduced into the National Immunisation Schedule in the US in 1999, and by 9 months, more than 1 million doses were given throughout the United States. And then they saw this. And this came up in the post-marketing surveillance, is episodes of interception. They were watching very carefully under the licensure, and when they put the vaccine into the population, more than 1 million doses, what they were seeing is that babies after their first dose, there was a little peak here of episodes of interception in the first 7 days after vaccination. Didn't occur so much with the second dose and hardly at all in the third dose. Now, as you know, interception is a common paediatric surgical condition. It occurs without vaccination, but this was seen as a temporal relationship to vaccination and did worry the FDA and the various regulators. And as a result of that vaccine was suspended, the recommendation was suspended 9 months after it was introduced. Subsequent studies have looked at what the risk of interception was with the RotaShield vaccine, and it's probably an episode of interception in 1 in 12,000 to 1 in 30,000 babies immunised. So not a common side effect. Certainly would not have been picked up in the clinical trials that had been done, which were 5,000 to 7,000 babies. However, interestingly, they did see in their clinical trials of 5,000, I think there was 3 in the vaccine arm and 1 in the placebo arm. But at the power of the calculation, couldn't assume that that was actually by chance, that such small numbers. And so on the product label, when the original licensure, it was said that they needed to follow up and be aware of interception as a possible risk. And that in fact happened. So this occurrence, being taken off the market in the US, was a huge blow to the development of rotavirus vaccines. Because although not many deaths occur in the US, quite a lot of hospitalisations, but this was considered an unacceptable risk in that setting. And up until that time, there had been no clinical trials really very much outside the US really to give evidence in high burden rotavirus areas where this risk may have been totally acceptable. This is a risk of interception that's not death from interception. This is just an episode presenting. So the RotaShield vaccine went successfully through the different phases into licensure, but fell over at the post-marketing surveillance. So even though you think you've got a product that's going to solve the problem, vigilance and the importance of surveillance systems to pick up rare and unacceptable adverse events is incredibly important. So back to our translation, back to our lab. The problem still exists in the community. The world, is still needs a rotavirus vaccine. You'll be aware that there are now 2 commercial rotavirus vaccines have been developed since that time. The RotaTeq vaccine from Merck and the Rotarix vaccine from GSK. They're both developed on quite different virological principles. The RotaTeq vaccine, a bit similar to the original RotaShield vaccine, it's got an animal component from bovine, so cow, with a component from human. In comparison, the Rotarix vaccine is a single rotavirus that has been developed from a human rotavirus, from a child that had diarrhoea. And so that has been developed as a single rotavirus vaccine with a single serotype. These vaccines have gone through all the various phases. Because of the experience with interception at a possible 1 in 30,000 for the RotaShield vaccine, the clinical trials of the two vaccines now, RotaShield and Rotarix and Rotatec, have been the largest clinical trials ever performed, with the clinical trials of both of those vaccines in over 65,000 babies. All to prove that the risk of interception with these two vaccines was not greater than in the RotaShield vaccine of 1 in 30,000, because this is placebo controlled. So half of the babies will receive placebo and half active vaccine. In addition, because of the interception problem, there were large post-marketing surveillance studies that were required in many of the early introducer countries. Because of the success and the evidence of efficacy with these two vaccines, WHO provided a recommendation that all children be vaccinated. A number of tools were developed to assist countries that were introducing rotavirus vaccines to ensure that there was safety associated with introduction, and to provide guidance on what sort of safety, surveillance and pharmacovigilance were required in those countries if they really wanted to understand either any rare event, but in particular interception. So this is just a little bit of an overview. The two vaccines currently licensed for global use and implemented now in 75 countries around the world are very effective. They're two different, as I say, different principles in the virology. One a pentavalent bovine reassortant and the other is a monovalent human. They're administered in either 3 doses or 2 doses for the Rotarix. The clinical trials for safety are very similar, and there is a very small risk of interception in some countries but not in other countries. In Australia we've done quite a lot of post-marketing surveillance for the Rotarix and Rotatec vaccines, and we see an incidence of interception around 1 in 50,000 to 1 in 100,000. The efficacy against severe rotavirus gastroenteritis is excellent in most countries, somewhere around 98 or 85 to 96% in developed countries. And the efficacy against severe diarrhoea in children less than 1 year, any diarrhoea, is also very good. So as I mentioned, 75 countries as of January this year have introduced rotavirus vaccines into their national immunisation program. But there's many grey areas, including Indonesia, that have not as yet. In countries that have reduced rotavirus vaccines, there's been a lot of work around epidemiology and understanding the effectiveness of vaccines. This is completing the cycle. Introducing the vaccines, do the vaccines work in the real world? Not only are they safe, but do they work? Do they provide the result that was intended? Well, the reduction in hospitalisation in children less than 5 years, or less than 2 years and less than 5 years, but this data is less than 2 years, has been very effective. In Australia, 87% reduction of rotavirus-associated hospitalisations after introduction of the rotavirus vaccine. So they work. Interestingly, they work also for all-cause diarrhoea hospitalisations by about 20% in a number of countries as well. And that might be just because we don't know the cause of some all-cause diarrhoea, or there may be secondary infection that may influence this as well, that the babies may get a secondary bacterial infection after a rotavirus infection. If you interfere with that process, you may reduce vaccination stress, or with vaccination you may reduce this. We're also seeing early data now about reduction in deaths following introduction of a rotavirus vaccine into the national immunisation program. This is data from Mexico. They were also seeing it in Brazil, both middle-income countries with a significant rotavirus disease burden. And certainly Mexico, 41% reduction from all-cause diarrhoea. The information on safety profiles suggests a very good safety profile, although there is a low risk of interception. And there is a general recommendation from WHO and most regulators, certainly our Australian regulator, is the risk of interception is so small and so rare, but the benefits are so great that, our regulator and certainly WHO are recommending vaccination. So have we solved the problem? Is it all, why on earth are we doing any more studies of rotavirus disease now? We've worked in the lab, we've got a vaccine, 2 vaccine candidates, they've gone through the whole process, they've had extensive post-marketing surveillance, and we've completed the cycle. We know that they're effective and safe. Not quite yet. Not quite yet. What has been a bit disappointing with rotavirus vaccines have been that the protection offered in developed countries, like Australia and US and Europe, is very high. But the protection offered in less well-developed countries, low resource and middle resource countries, has been really disappointing. And in some countries like India, Bangladesh, Pakistan, Vietnam, the efficacies of these vaccines are 50% or so, 55%. It means that you're protecting half the children from severe rotavirus gastroenteritis, but it's only half as good as we hoped, and not as good as what is happening in developed countries. And there are a number of reasons that could be postulated. One is the environmental difference. The high breastfeeding rates may interfere with uptake of vaccine. The intestinal flora of babies might be different. There's a variety of other postulates, none of which are completely known at this time. There are other barriers to introduction of rotavirus vaccines in low-income countries, cost being an important one. In Australia, the cost of the rotavirus vaccines is somewhere between $120, $180 Australian for a course. So it's a lot of money. On the private market in the US, it's more than that. Within a national immunisation program, it's sustainable. Gavi countries have got a subsidy for this, but it still costs countries money. These vaccines are expensive. So that there is cost to this important limitation, also the need for the cold chain, which would be nice to avoid if we possibly could, particularly in remote areas where the cold chain can be very variable and transportation challenging. We also questioned or asked the question, is it possible to protect babies from rotavirus disease from birth? The current rotavirus vaccines can only be given from 6 weeks of age at the very earliest. And so there's a period from birth to 6 weeks where babies are not protected and can't be protected on the current vaccines. We know in low-income countries the epidemiology of rotavirus suggests that rotavirus infection can occur very early, and these children are vulnerable. So if we're going to start again and improve things, go back to the cycle, what improvements can we make to a current rotavirus vaccine? And I'd suggest if we can give a birth dose of rotavirus vaccine, a cheap vaccine, a vaccine that's going to be effective in countries where the disease mortality and morbidity of rotavirus is very high, so overcome that 40 to 50%, it would be ideal and really would be starting to really challenge and meet the dream of rotavirus vaccines had to offer. So this is the work that's being done here in collaboration from the team here at UGM and Murdoch Children's Research Institute in Melbourne with Biopharma in Indonesia, University of Otago in New Zealand, and they're the major collaborators, developing the RV3 rotavirus vaccine to look at a very unique neonatal rotavirus vaccine to protect rotavirus disease from birth. And this really aims to address the lower efficacy in developing countries. We believe if we can put the vaccine in at birth before the mothers are established breastfeeding, before the infant gut has developed its own microbiome and a challenge to vaccine take. And get it in there, establishing protection early during that gap period, we might improve that efficacy in developing countries. Also, at birth is a time when mums and their babies are often in a healthcare setting, so it's easier to implement a vaccine and ensure that at least babies go home with one dose of vaccine, particularly in remote areas where vaccine coverage might be quite variable. It might address safety concerns because intussusception is very rare in the first days of life. It usually starts to be a problem maybe around 6 to 8 months of life when it starts to the natural history of intussusception. We would like to address the cost issues, vaccine supply, and we believe actually the ideal timing to deliver a vaccine is at birth. We've gone back to the lab and looked at a specific vaccine that might be modelled using nature, really, to take advantage of nature. And I mentioned a little earlier that neonatal rotaviruses are naturally quite unusual and quite different to other rotaviruses that cause disease. Newborn babies, probably in Yogyakarta, certainly in Melbourne, often are infected by a rotavirus which doesn't cause disease but replicates very well in the infant gut. And it's believed that these neonatal rotaviruses might, by their specific configuration, attach to specific receptors in the newborn gut which aren't present in later gut. And that they're able to produce, and there are some variety of different laboratory explanations around the differences in structural proteins of these particular human neonatal rotaviruses. RV3 is one of these human neonatal rotaviruses. It was isolated from a healthy newborn infant in Melbourne. Graham Barnes and Ruth Bishop followed these babies that had this natural infection with rotavirus for the first 3 years of their life and found that the babies that had been affected in the nursery with this RV3 strain were protected from severe rotavirus gastroenteritis for the first 3 years of life. The babies were infected but did not develop disease compared to those babies who did not have that infection in the neonatal nursery. And this particular strain appears to be very stable, so we don't believe it's vulnerable to change. So we've taken advantage of what nature has been saying to us around neonatal natural infection in nurseries and developed that one of the strains from a newborn baby into the RV3 vaccine. And so this vaccine is developed from that single human neonatal strain. It's monovalent, very similar to the Rotarix vaccine viral strategy program. It targets birth dose delivery because we know this is very safe in newborns. The newborns that had this natural infection were very well and had no problems. You would not have known that they were excreting and had been infected with this. And we have believe that could help with some of those challenges of current rotavirus vaccines. However, we have to go along the pathway of all vaccines and prove that the vaccine is safe, produces an immune response and is effective. So the phase 1 studies in Melbourne, again, as I mentioned, they really are looking at the tolerance to vaccines. This phase 1 study, we did it in adults first, proved it was safe, did it in children 3 to 8 years, and then did it in infants 6 to 8 weeks of age, looking at 10 in each cohort against placebo. And it was well tolerated in all those groups, and we had some evidence, early evidence of vaccine take in the infants. So we ticked the first box, phase 1 trial. We've just completed a phase 2A safety and immunogenicity study in New Zealand, in Dunedin. And that was to look at the vaccine take, looking at immunogenicity, vaccine take, following administration of 3 doses of the vaccine, whether it was given with the first dose as a newborn dose, or the first dose followed by 2 doses, or the first dose as an infant dose at 6 to 8 weeks of age, like the other vaccines, followed by 3 doses. And we looked at various safety issues and in addition to immunogenicity. And the study was completed last year, and we've just had the results coming through. And what it shows in these two bars, this is the neonatal schedule with the first dose given at birth, followed by 2 doses, or the infant schedule with the first dose given at 6 to 8 weeks, versus placebo. And what we show here is the vaccine take was very good in both of the arms where the vaccine was given compared to placebo. And these placebos had a natural wild-type infection, as you might expect babies when they're starting to be 4 months of age. So this suggested and provided good evidence that the vaccine actually could support a very strong and robust immune response in over 90% of babies after 3 doses. So we showed it was safe, associated with strong vaccine take, and provided an opportunity for early protection with the birth dose. So we've got number 2, tick. Phase 2, tick. So coming to the trial now that has been conducted here in Yogyakarta. This is a safety, immunogenicity and efficacy study. So we're looking now at protection in addition. So it's very similar to a phase 3 trial. The reason it was called a phase 2B, at the time when we started we had a small sample size that I'll explain to you in a moment. We've actually increased that sample size, so it's actually really a phase 2B/3 because we're looking at efficacy and protection. And this study is a double-blind, randomised, placebo-controlled trial. It started in 2 provinces here and commenced in January 2000. And I'm saying, there's an old slide. 100% recruited as of January. Why develop this type of vaccine, particularly for Indonesia? Well, there are a lot of deaths, as I mentioned, and the current in Indonesia, and the current vaccines are pretty expensive for inclusion into the National Immunisation Program. And you have a very good manufacturer, Biopharma, who are willing to take this challenge on board and produce an Indonesian rotavirus vaccine. So we've been working in the field. We've graduated our first participant with this study because it's looking at protection. We the child has 3 doses of active vaccine, 4 doses of investigational product. I won't go into the study design. But the child is followed up for 18 months, until they're 18 months of age. Because what we want to know is whether they're protected during that 18 months of age from gastroenteritis. So that first little baby was graduated, so 12 months of age. That's fabulous, back in June. We've got a great team working really diligently and hard. This is the first baby that was dosed at the launch, the official launch. The recruitment was completed, a wonderful milestone in January of this year. 100% recruitment of the first cohort of 1,649 babies, 1,649 babies, a huge effort. It's not only recruiting these babies that's important, following them up. They're followed up weekly. Each episode of diarrhoea is captured, stools are collected, huge amount of documentation, because the aim that this will be an important trial for the submission to BPOM. This trial is monitored by an external group to make sure that every aspect of the trial is documented and done according to the protocol, and all the adverse events and any concerns are picked up and documented and verified. It's a huge effort, and it's a different way of running a clinical trial as an academic, to have someone over your shoulder, but it's an essential part that regulators require you to do if you're going to use these trials to support a submission to a regulator. So it's been a huge effort by a number of primary health cares in Central Java and Yogyakarta region, Kelantan, Sleman region, who have consistently recruited over this period of time and continue to follow these babies up. Although the recruitment has finished, they've got to follow these babies up until 18 months of age. Lots of documentation. And we sometimes wonder why there should be so much, but it's, as I say, it's required. Because there is seen to be much promise in the RV3 vaccine, not only for Indonesia, but potentially addressing a major global concern. We've been asked collectively to accelerate the development of the vaccine as quickly as we can, and to that end, we're just about to embark on the expansion of the sample size to bring it from a 2B study towards a phase 3 study, and to add more babies into this trial to really get a good, strong efficacy endpoint for the RV3 vaccine. So we're moving to almost 3,500 babies. Although the recruitment has finished into this, the first cohort, we've got a little bit of a break to tidy up and get everything ready, and then we're going to start again to recruit into this trial further. So this is where we are now, coming up towards the phase 2B3 phase of this program. And you know, we're starting to see the light at the end of the tunnel, which is great. Biopharma are working very diligently. I haven't spent a long time talking about their vaccine development, but parallel to them, to the clinical trial development process here, they are working on developing their own rotavirus vaccine and increasing the yield, trying to reduce the costs. They've got some innovative ways. They want to produce the vaccine with some bovine trypsin, which will make it a very unique vaccine. And so Biopharma are, as I say, working very hard to be able to make the vaccine efficiently, effectively and rapidly, so that hopefully by the time we have finished this trial, all the documents and all the work will line up, and we will be able to submit to the Indonesian regulator for the Biopharma produced RV3 vaccine. Completing the cycle. At the end of that, we will have to go back and assess again post-marketing surveillance. This is the cycle until you think you have something perfectly done. But I would say perhaps the secret to research collaboration exists on part of that cycle, having a significant, important disease to focus on, that there is a good aim for your study and a good aim and intention for what you're about to produce. Strong science from the laboratory to the clinic to the public health, strong science to support, and a great collaborative team. It's fun to work with. That helped. So thank you very much.